So let's see the cycle of glycolysis in respiration in plants. Initially, sucrose. Sucrose is converted to glucose plus. So glucose plus fructose. So actually this fructose is also converted to glucose by invertase. So this is the enzyme given even in the NCERT textbook also. Now let's start the glucose. Because this chapter is respiration in plants. So that's why we have to see from the perspective of plants only. Next, glucose is converted to glucose 6-phosphate by the enzyme called hexokinase. And here one ATP is utilized to form ADP plus PI. And glucose 6-phosphate is converted to fructose 6-phosphate by the enzyme isomerase. And now fructose 6-phosphate is converted to fructose 1,6 bisphosphate. Here one ATP is used to form ADP plus PA. So the enzyme is phosphofructokinase. Right? Now fructose 1,6 bisphosphate is splitted into two things by aldolases. Glyceraldehyde 3-phosphate and dihydroxy acetone phosphate. Right? And here the enzyme, it is a reversible reaction. Here the enzyme is isomerase. And next, now we got 2 into glyceraldehyde 3-phosphate. Now it is converted to 2 into 1,3 bisphosphoglycerate or glyceric acid. Here 2 NADH2 is produced from NAD plus. So the enzyme is glyceraldehyde 3-phosphate dehydrogenase. So I am not writing the entire enzyme name. 1,3 bisphosphoglycerate is converted to 3-phosphoglycerate. So here the enzyme is, so here actually 2 ATPs are produced from ADP plus PI. So if you see here the enzyme is phosphoglycerokinase. 3-phosphoglycerate is converted to 2-phosphoglycerate. Here the enzyme is phosphoglyceromutase. and 2-phosphoglycerate is converted to PEP, phosphoenol pyruvic acid. Here 2 ATP are produced from ADP plus PI, and the enzyme is enolase, and finally PEP is converted to pyruvic acid or pyruvate. Excuse me for this. Actually here no ATP is produced, and the ATP that is produced is here. 2 ATP are produced from ADP plus PI, right? So here the enzyme is pyruvate synthase, or you can also have pyruvate kinase also. So what is the total number of ATP is here is, if you see here, these two are utilized. So 2 ATP utilized, and if you see here 2 ATP are produced, totally 4 ATP are produced. So 2 ATP utilized, so the net will be 2 ATP of partial oxidation. Now we talked about partial oxidation or glycolysis or EMP pathway, Embden-Meyerhof-Paranas pathway. So let's see about the next thing. What happens to this pyruvic acid? We will be having 2 pyruvic acid. So now this pyruvic acid will be going to mitochondrial matrix. And there this pyruvic acid is converted to plus coenzyme A plus NAD plus, gives rise to acetyl CoA plus CO2 plus NADH2. Here the enzyme is pyruvate dehydrogenase, and now we are entering into the Krebs cycle. So now acetyl CoA plus OAA. Acetyl CoA is 2 carbon compound, OAA is 4 carbon compound. Acetyl CoA and OAA are combined to form citric acid. So here 2 acetyl CoA is formed. Okay, citric acid. Here the enzyme is citrate synthase. This is 6 carbon compound. Now afterwards the citric acid is converted to cis aconitic acid. This is also 6 carbon compound. Cis aconitic acid is converted to isocitrate. This is also 6 carbon compound. Isocitrate is converted to alpha ketoglutaric acid. This is 5 carbon compound, and here NADH2 is produced. Actually 1 carbon dioxide is gone and NADH2 is produced from NAD+. And now alpha ketoglutaric acid is converted to succinyl CoA. Here also this is 5 carbon compound, this is 4 carbon compound, 1 carbon dioxide is gone, and here NADH2 is produced from NAD plus, and now succinyl CoA is converted to succinate. Here one GTP is produced from GDP plus PI, and succinate is converted to fumarate. So fumarate, here FADH2 is produced from FAD plus, fumarate to malate, malate to oxaloacetic acid. Here also one NADH2 is produced from NAD plus. So if you see here, we have 2 ATP here, that is for sure. We have 2 ATP from glycolysis. From glycolysis, here we have 2 NADH2. Okay, so 2 NADH2. And coming to the link reaction, here we have 2 NADH2, totally 4. And this cycle will happen 2 times because 2 acetyl CoA is formed. So this NADH2, 2 more 6, 2 more 8, 2 more 10. So totally 10 NADH2 is produced, and 2 FADH2 is produced, 2 GTP is produced, 2 ATP is produced in glycolysis. Okay, so let's see for 1 NADH2. For 1 NADH2, 3 ATP are produced. For 1 FADH2, 2 ATP are produced. 1 GTP is equal to 1 ATP. So totally if you see the balance sheet, 30 ATP, 4 ATP, 2 ATP. So if you see here, from Krebs cycle we are getting 36 ATP plus 1 ATP, plus 2 ATP from glycolysis. So totally 38 ATP. How this 38 ATP are generated, we are going to see. That is ETS. So before going to the ETS, we have to draw this complexes. So let's name it. Here if you see, this is inner mitochondrial membrane. This is matrix. This is intermembrane space. So let's see the complex names. This is complex 1, 3, 2, 4 and 5. So complex 1. Complex 1, 2, 3, 4 and 5. The name of complex 1 is NADH dehydrogenase. Complex 2 is succinate dehydrogenase. And complex 3 is cytochrome BC1 complex. And fourth complex is cytochrome C oxidase. Fifth complex is ATP synthase. We'll write the subcomponents here. FMN, flavin mononucleotide, and we have the iron sulfur complex. Here it is second complex, flavin adenine dinucleotide, iron sulfur complex. Here it is cytochrome C, FES in between, site B1. And here it is, for this you remember, for cytochrome C oxidase you will be having copper A, two copper complexes, copper A and copper B, site A, site A3. So you have to write like copper A, site A, site A3, and we have the copper B, ATP synthase F0, F1. So let's see here, NADH2. So we are removing the electrons. 2 electrons we are removing from this, and we are converting this into NAD plus, and 2 protons are removed here. Okay, we remove 2 electrons at the same time, 2 protons. Now the 2 electrons are being moving from FMN to FES, FES2 through mobile carrier UQ, we call it as ubiquinol. By adding electrons, it will be converted to ubiquinol. When electrons is being taken, it will take protons also. So now it is going to site C. Now from here, succinate dehydrogenase complex, FADH2, is oxidized as FAD plus. Now the protons are released here, and the 2 electrons are been going from FADH2 to FAD plus. So FAD to FES, FES2 through same UQ, mobile carrier, it will become ubiquinol. Ubiquinone will become ubiquinol. Ubiquinone to H2 will become ubiquinol, and this is site C. So from here, actually 1 electron will be out of here. We are giving 2 electrons. Here also we are giving 2 electrons, but 1 electron will stay here itself. So 1 electron will be here, 1 electron will be here. So totally the remaining 1 electron and remaining 1 electron will move through site C to iron sulfur, site B, from site B to site C, site B1 to site C, to the fourth complex. From the fourth complex, the 2 electrons will move to, because here from the 2 electrons 1 electron is stayed here. Here from the 2 electrons 1 electron is stayed here, and remaining 1 electron is moving here as 2 electrons. So site copper center A to site A, site A3, copper center B. Finally it is given the electron acceptor is oxygen, plus 2H plus gives rise to H2O. Now the oxygen is the final electron acceptor. It is being formed as water. So now how the ATP is produced? Here they have split into, if you see, the 2 protons, and when electrons is being taken to the iron sulfur complex, 1 more 2 protons will come into the intermembrane space. Totally you will be getting of 4 protons. And here if you see, from the complex 3 also you will be getting 4 protons, because we have 2 iron sulfur complexes here, so 4 protons. And from this, for giving 2 electrons in an exchange, the system is taking 2 protons. So totally if you see, you have 10 protons. For every 2 protons, so for all these things, here the concentration of protons is increased in the intermembrane space. So for every 2 protons, 1 ATP is generated. So 2 protons. one ATP is generated. So ADP plus PI gives rise to ATP. So like that, assume that for NADH2, the electrons are moving from center 1 to 2. So if you see, so usually for our understanding they have given 1, 3, 2, 4, 5. How the NADH2 is been producing 3 ATP. For every 2 protons, what we told, we are getting 1 ATP. So for NADH2 we will be getting 3 ATP. How is this? Now first complex name is NADH dehydrogenase. Now the electrons are moving from 1 to 2. So 2 protons we are getting for 2 electrons. And now from 2 to 3, 2 protons we are getting here. And from 3 to 4, 2 protons we are getting. Totally how many? 6 protons. For every 2 protons, 1 ATP. So totally 3 ATPs for NADH2. For FADH2, if you take 2 to 3, 2 protons. 3 to 4, 2 protons. So totally 4 protons for FADH2. So 2 ATP. Done. This is the ETS complex. So coming to the next thing, amphibolic pathway. In the amphibolic pathway, if you see, glucose. So we will see the respiratory substrate, amphibolic pathway in the sense. So Krebs cycle will be act as a catabolic as well as anabolic. Fats, proteins. So let's see. Fats are split into 2 compounds. One is fatty acid, other one is glycerol. And proteins are split into amino acids. So glucose is forming final complete steps as dihydroxy acetone. So this will be converted as glyceraldehyde 3-phosphate. So now the glycerol will be converted to dihydroxyacetone, and from that glycerol it will be converted to glyceraldehyde 3-phosphate. Like that the reaction will be happen. So if the glyceraldehyde is converted to pyruvate, so amino acids will be forming as pyruvate by the process of deamination, and pyruvate is converted to acetyl CoA. So fatty acid is converted to acetyl CoA. Acetyl CoA is we will be going to Krebs cycle. So carbon dioxide is removed and it will go to the ETS. Water is formed. So this is amphibolic pathway, because you can take the acetyl CoA for the preparation of fatty acid. You can take the glyceraldehyde 3-phosphate for the preparation of glycerol. So we can consider the pyruvate for the preparation of amino acids by amination and later proteins. So like this we can make it as amphibolic pathway. And next we will be having the other thing, that is RQ, respiratory quotient. So carbon dioxide produced, the amount of carbon dioxide produced by amount of oxygen utilized. So remember one very important mnemonic, carbon dioxide, carbon means 1, okay, so 1, so equal 6 by 6, 1. Fats, so write this F in this way, 7, 0.7. Proteins, write P in reverse, 0.9. And next, xerophytes or succulents, 0. They don't produce any carbon dioxide. So 0 by, assume that 0 by 27. So 0 by anything is 0. And in case of organic acid, infinity. Assume that 27 by 0. It would be infinity only. Thank you.